Hey team, Jack here. Today I'm going to take you through a really cool scientific technique called Flowcytometry. I dabbled on a little bit in the previous video, but now I'm going to go a little bit deeper. This image here is actually inside a flow cytometry machine, and one of the defining features of a flow cytometry machine is the lasers. And every flow cytometry scientist likes to brag about how many lasers and how many colors and how many detectors their flow cytometry machine has, so they can look at a million things simultaneously, right? I never have gone past maybe 5 things and simultaneously, but people love to go up to 40 or 50 different cell types and cytokines and so on. Anyway, so this is a beautiful flow cytometry because it's got so many lasers. Alrighty, so let's jump into it. This is what they look like with less cool imaging. And essentially, you've just got a place to administer the liquid, and then it goes into this black box of lasers. And we're gonna crack that open and have a look in at what that looks like in reality. So you can either use a 96 well plate, or you can use what's called a fax tube. And essentially, a tube comes out of the flow cytometry machine and sucks up the liquid. If it's using a 96 well plate, it actually has the ability to go from well to well. Now, a very important thing is it's going to suck up the liquid that contains the cells, right? And in between wells in your 96 well plate, it's going to want to clean the pipes. So all flow cytometry machines are hooked up to big tanks of cleaning solution, basically a buffer with a little bit of detergent in it, which helps flush the lines before it starts going to the next cell, where it will start sucking up the cells. Right, so it sucks up the cells, and then it's going to shoot them through a very narrow gap. So they're going to go boom, 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 down a very narrow gap, where only one cell can go at a time, pretty much. And then so we can investigate and probe each cell one at a time. So how do we probe them? Well, we probe them with those fancy lasers and those fancy detectors. Now, in the previous video, because it wasn't about flow, in the previous CBA video that I did, I brushed over what forward scattering was, but now I'm just going to give you a bit more of a deep dive on what forward scatter is and why it's actually called forward scatter. So the cell comes down here and it will get hit with a white light. Now this white light will hit the cell. Now a good portion of that light will go straight through, and what the flow cytometry machine does, it actually blocks the light that goes straight through. It doesn't want to know what light gets straight through, because that's going to overwhelm the signal, because cells are mostly transparent, right? So most of the light will get through and it will overwhelm the sensor. So it has this little blocker there that blocks the direct light getting through. But some of the light, depending on the size of the cell, is scattered forward. So it's slightly deflected, so it misses this blocker and is now picked up by the sensor. And so this is what we call forward scatter. So it's the light that is scattered going forward, and it largely dictates cell size. The bigger the cell, the more scattering, the more forward scatter you're going to get. So the forward scatter is roughly picking up the cell size. But there's another kind of scatter that we're also going to pick up at this laser. We're also going to pick up side scatter. So this is 90 degrees to the forward scatter, and this is light that has hit the cell and bounced off perpendicular and has been picked up by this sensor. Now this is called side scatter, and it typically measures complexity and granularity. Now a way I like to think about it is, this is a regular boring cell, it's a glass orb, and most of the light goes through and a little bit of it gets bent a little bit, so it's going to have some forward scatter, but it's not going to have any side scatter. The light's going to go mostly straight through. Whereas this is a granular side, this is the complex cell that's full of granules. The light will go in, and because of the complexity and the granules inside the cell, the light's going to bounce off in all directions like a disco ball. And actually, if you look at the cells, you can kind of see. So this is a monocyte. It's not going to have a huge side scatter. And this is a basophil. It's full of granules. It is going to have a lot of side scatter. It's the disco ball inside your blood. So that includes eosinophils and neutrophils, whereas lymphocytes are much more like this glass orb. So that's how side scatter works and how forward scatter works. But the real crux of a flow cytometry is fluorescence. It loves to detect fluorescent labels. So what we do is we put a fluorescent label on the antibody, and that antibody will only bind to a specific protein, and that protein is something that we're interested in. So let's say this is bound to the T cell receptor. It's a T cell receptor antibody. It will bind to this cell because the cell is a T cell, but it hasn't bound to these cells. So maybe these cells are B cells. So this antibody, because it's only binding to the T cell receptor, is now distinguishing between a T cell and a B cell, or a T cell and any other cell, a monocyte, for example. So it will go down that cell and we will get the forward scatter and side scatter information from that cell. And then with that same cell, it will go down to a different laser. And this laser will emit, for example, a blue light, and this blue light will hit the fluorophore, and the fluorophore will change the wavelength of that light to green, and this will now go through a filter. So this filter will filter out the blue light. So the blue light coming straight from the laser will not get through to the sensor. So any sort of blue light just coming through is blocked. But the green light will go through the filter and hit the sensor. So only a cell with this antibody will have the fluorophore, and that fluorophore changes the color of the light that hits it and then is emitted, and only that color change will be detected. So any cell without that antibody, therefore without the TCR, therefore without the fluorophore, will not be detected and will not get that green signal from that cell. Now, flow cytometry machines often use a single laser source or multiple laser sources. And also, instead of using a filter, they often use a special mirror. And this mirror will only reflect a certain wavelength and will let all the other ones pass through. But it's essentially doing that same thing. You see, all the blue light is filtered out here, and so the other light can get through, and then all the green light here is reflected. So our cell from the previous slide will be detected on this sensor, and only this sensor. It won't be detected by the other laser lights, the other detectors there, because the other light will be going straight through the mirror and it won't be reflected. So instead of having a filter in flow cytometry machines, you often have a mirror that's special in that it can only reflect a single wavelength. And so now we put it on graphs like this. So on the y-axis, we've got forward scatter, which corresponds roughly to cell size. And on the x-axis, we've got fluorescence. And in this case, I'm saying that the antibody is for the MHC2 molecule. Now, the MHC2 molecule is on antigen-presenting cells like macrophages, for example. So we get the cell up here. Now, this cell is large and is positive for MHC2, so we could probably guess that that's a macrophage. Macro meaning big, and it's got MHC2, so that's a macrophage. Down here, we've got a small cell, not fluorescent. It's small and it's not fluorescent, so this could be any number of cells. It could be a B cell. No, it could be a T cell. B cells have MHC2, sorry. So that could be a T cell down there. And then over here, we've got a small cell with an MHC2 receptor. So this could be a B cell or it could be a monocyte, for example, because this is a small cell that's positive for MHC2. It's highly fluorescent, but it's small. And so you can see we get cell populations here. And this is the beauty of flow cytometry. We can start to quantify how many of each of these cell types we've got in the tissue or the blood or whatever we're looking at. So in this example here, perhaps they took some bone marrow. They took some bone marrow here. They turned it into a single cell suspension. They then put an antibody for MHC2 in, and then they looked at the forward scatter and the MHC2 fluorescence. And you can see we've got these populations here. We've got MHC2 negative cells. They're very low, so they're negative and they're small. Then we've got small MHC2 positive cells. This could be B cells, could be monocytes. And then we have MHC2 positive cells that are large. So these are probably macrophages. And what we can do in the software, so this is a classic heat plot from flow cytometry. The color change represents overlapping cells. So as you get more and more cells sitting on top of each other, you'll get closer and closer to red. So there's a lot of cells there all sitting on top of each other. And what's cool is you can draw a box around it and say what percentage of all the cells are these cells. And we can start to quantify 2.7% of these cells are probably macrophages because we've drawn a box around these. They're MHC2 positive and they're large, right? So these 2.7% of our cells are macrophages. So now let's run through a full flow cytometry, right? Let's imagine this is a tube I've got a tube of cells here, and here I've got a small cell that might be a T cell, I've got a big cell that might be a macrophage, and over here I've got a dead cell. You can see that there's holes in the membrane. This cell has died, right? So let's see, what would we do as flow cytometry researchers? Well, the first thing we'd do is we'd put in an antibody that will bind to a particular receptor that we're interested in. So let's say this is the T cell receptor here, and we've got an antibody that will only bind to the T cell receptor, and it's got a green fluorophore on it. So now this T cell has been successfully labeled green. So now we could distinguish this cell from these two cells because it's green, meaning that's the T cell. We then centrifuge it, and ideally we balance the centrifuge. This is an ultra centrifuge. They get up to 100,000 Gs. That wasn't balanced, and so that internal metal got out of balance and then just spun so quickly that it smashed itself into bits. Really, really cool. So definitely balance your centrifuge there, and that brings all the cells to the bottom. So now they form a pellet, and now we can remove the liquid and put in a new liquid, right? So that's why we centrifuge. And so what we might do next is wash to get rid of all of the unbound antibodies. So we'll resuspend in a wash buffer, then centrifuge back down again, then remove that liquid. And we might do that a couple of times. And then the next time we might resuspend the cells in a liquid that contains an antibody for, say, over here, a macrophage membrane protein. In this case, it might be MHC2, right? So now this one will have a different colored fluorescent label on it. So we've got a green antibody bound to the T cell receptor, and we've got a yellow antibody bound to the macrophage receptor, the MHC2, the antigen presenting molecule of macrophages and a bunch of other cells. But in this case, it's going to just label the macrophage. And so now we're distinguishing between these two cell types. How do we distinguish between this cell type though? So we do another centrifuge, remove that antibody liquid, maybe wash it a few times, and now we put in a dye. Now, this is a special kind of fluorescent dye. One example is propylium iodide, and this dye cannot get into cells unless they have a permeable membrane, right? So it can't get inside a cell unless there's a permeable membrane. And another unique thing about this dye is that it doesn't fluoresce until it binds to DNA. So it's not fluorescent, but once it binds to the DNA, then it turns on and becomes fluorescent. So if we resuspend our propylium iodide solution and we put that in here, it's going to go into only the dead cell and make it fluoresce red. So not all fluorescent labels in flow cytometry are antibodies. Some of them are to distinguish between cells, for example, that are live or dead using these kind of live dead stains such as propylium iodide. So now we've got three different colors here in our cell populations, and we'll be able to distinguish T cells, MHC2 expressing cells, and dead cells, all by running our flow cytometry plot. So here's where things get a bit complicated. We've got a lot of lasers, a lot of colors, forward scatter, side scatter, different antibodies, so on and so forth. How do we sort of collate that data? And what we do traditionally is we call what's called a gating strategy. And this is where we can sort of break down the populations bit by bit to bring out some coherent data using what's called a gating strategy. So let me take you through one. So in the point of this gating strategy is to answer this question. What percentage of our T cells are cytotoxic T cells? And what are T helper cells, right? So we're going to distinguish between those two T cell populations using flow cytometry, and we want to answer this question. So the first thing we do is we've got forward scatter and we've got side scatter. So these cells here have a lot of side scatter and forward scatter. These have not a lot of forward scatter and side scatter. So these are small, uncomplicated cells. So we know that that's where our lymphocytes are. Lymphocytes are small and uncomplicated. Up here is our granular sites like neutrophils, eosinophils, and so on and so forth. So these are our non-granule cells. So we draw a box around them. So we've drawn this circle around them. I sometimes call it the circle of trust because you have to trust the researchers to draw the circles in the right place. So I've drawn a circle around it. And then I'm going to say, I want you to take only these cells. It's a gate, right? Only let these cells through the gate into my next analysis. So of these cells, can we graph a different bit of information from it? So we've taken those cells over to here, and now we've said, okay, how much do they fluoresce on the dead cell stain? Remember, propydium iodide that will bind to the DNA only if there's holes in the membrane of the cell. And we've just got forward scatter in here to track size. And you can roughly see that this distribution is very similar to this distribution. And that's because this is forward scatter, this is forward scatter, and these cells are those cells. But what we can see here is we've got some highly fluorescent cells on the dead cell stain axis, right? So this axis is measuring fluorescence. It's actually a log scale, so this difference is quite a big difference. And we can see over here, we've got a few cells here that are fluorescing the dead stain. So we can then draw another box around them and say, actually, we only want to look at the live cells. And we actually get a bit of information up here. Viable cells, 96.7%. So 96% of the cells are inside this box, and, 3% odd are outside the box here, and they have died. So we're already starting to collect information over here, right? And now we say we want to look at those cells and let them through the gate into our next graph. So the next graph is over here, and of these cells, we would like to know different labels here. So we've got CD45 and CD3. So here we've got fluorophores of certain colors. These have got to be different colors. So this might be orange and this might be green. We've got fluorophores for an anti-CD3 and an anti-CD45 antibody. Now, if you've watched all my previous videos, you'll know CD3 is an essential activator of the T cell receptor, and CD45 is an inhibitor of the T cell receptor. So if a cell has both the CD3 and CD45, it has an active functioning T cell receptor, and it's got both the inhibitors and activators of it that's critical to the function of the T cell receptor. They work as a unit together. So if you are positive for both of these, you are definitely a T cell. So you can draw a box around that. These are our T cells. These are not T cells, right? So these are our T cells, and we're going to put another gate around that. Remember, we're trying to answer the question, what percentage of our T cells, our live T cells, what percentage of our live T cells are cytotoxic T cells versus T helper cells, right? How does that population break out? So we draw a box around that. That's a gate, and we're only going to let these cells through into our next analysis. So we're only letting these guys through the gate into our next analysis. And the next analysis over here is our CD4 versus CD8 analysis. So on the y-axis here, we've got an antibody staining for the CD4 receptor, and on the x-axis here, you've got an antibody staining for the CD8 receptor, right? And so we know that these molecules recognize the different MHC molecules. We know that this recognizes the MHC2 molecule. This recognizes the MHC1 molecule. So these are CD8 positive T cells. These are CD8 negative T cells and CD4 positive cells. So these are our helper cells, and these are our cytotoxic cells. Now, what you'll notice is that there are some cells out here that seem to be positive for both the molecules. And that's kind of what I was talking about with the rainbow analogy. There is a lot of overlap, and there are sneaky subpopulations of T cells here. And I'd love you to have a think and maybe a Google about what kind of T cell is negative in CD4 and CD8, because there's a bunch of T cells down here, and you can maybe go off and Google it to find out there is a small sneaky subpopulation of T cells that don't have the CD4 molecule or the CD8 molecule. So they're not T helper cells, and they're not cytotoxic T cells. So you might want to go and Google that. But this is how our gating strategy works, and we end up with our final answer here. So 65% of our T cells that are in this circle here are CD4 positive cells. So 65% are T helper cells. And over here, we've drawn another box around our cytotoxic T cells, and 30.5% of our cells are cytotoxic T cells. So we've answered that question using our gating strategy. And this is pretty much the old school way of doing flow cytometry. A lot of people still do it this way. But there's another way, and the other way is called the TsNE plot. Now, there's a really complicated bunch of statistics that goes on here. But essentially what it does is it takes all the information in simultaneously. It doesn't just limit itself to looking at 2 different measurements at a time. It can look at all the measurements simultaneously, all the antibodies, all the forward scatter, all the side scatter, the live dead stain, all simultaneously, and it breaks them up into subpopulations based on their similarity across all those measures, right? And it's called dimension reduction analysis. So it reduces the dimensionality of the data. And I'll go into a little bit how that works later. It's a very rough approximation of how it works later. But you can see on this one graph, we've got our B cells here, we've got our CD4 positive T cells, we've got our myeloid cells up here, we've got our CD8 cytotoxic T cells over here, we've got our natural killer cells here, and we can actually quantify all our populations and what percentages they are simultaneously on the single graph, right? So now we're sort of this advanced statistical method that does a dimensionality reduction and clustering analysis. We can avoid the gaining strategies. Now, I mentioned circle of trust, right? I mentioned that researchers draw those boxes and say anything inside that box is a T helper cell, right? And it's kind of a circle of trust. We have to trust that that person has drawn the box right, they haven't sort of fudged any margins or anything like that. And this kind of takes the human out of the analysis. The TsNE plot takes the human out of the analysis and allows the analysis to occur naturally in an unbiased statistical program, okay? So there is a lot of pros there. Now, science journals have sort of cottoned on to the fudgeability of those boxes, and so now they ask for much better controls, they ask for the raw data, they ask for the raw plots, so that the journals can sort of review the data before they publish it. And that's the good journals, and that's the immunology journals. Sort of the generalist journals won't know about that and maybe won't go through that extra step. And so a lot of people are starting to now lean towards this more automated process of TsNE. Now, I'm going to very quickly give you sort of a general idea of how it works. So here we've got, say, forward scatter, and here we've got CD3 antibody. Now, remember, CD3 is that activator of the TCR. So if you're positive for CD3, you're a T cell, and over here, we've got a very large cell that's negative. So maybe those are macrophages. But I've actually colored these here to show you that there's 2 populations of T cells in there. There's the cytotoxic T cell and the T helper cell. But because of the way we're graphing it, and we're only looking at 2 dimensions, we're only looking at one fluorescent label and forward scatter, we can't distinguish those populations. So what we can do is put another dimension in there. We can put it on a 3D graph, right? So this is now 3D. We've got a Z-axis, a Y-axis, and an X-axis. On the Y-axis, we've still got forward scatter. On the X-axis, we've still got that CD3 antibody, so that labels all T cells. But now on the Z-axis, we've got a CD8 positive antibody. And now we can distinguish between the yellow and orange cells, because these orange cells are in the foreground. That means that they're right down here on the Z-axis, right? They are there on the Z-axis, so they are negative for the CD8 molecule, which means these aren't cytotoxic T cells. They are probably T helper cells, right? So they are way down there. These cells are high on the CD8 antibody scale, and they are high on the CD3 antibody scale. So they are both CD3 positive, which means they're T cells, and CD8 positive, which means they're cytotoxic T cells. So these are cytotoxic T cells. And there's a little Z-axis there to show that they're still very small, right? So this is now a 3D graph. And, you know, this is just an example showing you how a 3D graph works. You've got those 3 axes now. And so now you can see that some dots will overlap, and then as the graph rotates, you can distinguish them. And that's what we've got going on here. As the graph rotated, and we can now see the Z-axis, we can distinguish those 3 populations, right? So now we've got this distinguishing. Let's reduce the dimensionality of it. We don't need those 3 dimensions anymore. We can delete our axes and replace them. See how the dots stay exactly where they are? This is kind of, we've taken that 3D graph and we've just smushed it down to a 2D graph again. And so now we've got our TsNE. They should actually say TsNE 1 and TsNE 2. But basically, we have reduced the dimensionality of the data. We've reduced the dimensionality of the data. And we have smushed it down back onto its 2 points. And you can see now the cells are clustering each in a different location on our two-dimensional map. Now imagine having another label and another label and another label. You go from a 3D graph to a 4D graph to a 5D graph. It starts to get out of comprehension of a human mind, and it starts to become a statistical reality, right? So it's a complicated automated statistical process that generates these clusters by reducing the dimensionality of the data. So in this example, I reduced it from 3 dimensions back down to 2. But it can actually reduce a 4-dimensional graph down to 2 and a 5-dimensional graph down to 2. So it's quite cool, but it takes the human out of it. We don't need any gates anymore. So now we know those are the cytotoxic T cells, those are the T helper cells, and over there are probably the macrophages. Yeah, so we can get these populations here like this. So this is how this graph was generated. It took a very it kept reducing the dimensionality of the data of all the different labels. So there would have been a B cell label, maybe it was an IgG label, for example, CD4 T cells, CD8 T cells, gamma delta T cells. Maybe that's a hint to the question I asked you just before. Myeloid cells and natural killer cells can all be distinguished on one graph without doing that complicated gating and without human hands touching it, which is good. As you can tell, I'm pro this. There are still a lot of people that are anti this and pro gating. So, you know, there's a bit of debate in the research. I'm all for this, I think. Okay, now, what if you wanted to stain cytokines? So far I've only talked about proteins that are on the surface of the cell. But what if you wanted to stain a cytokine? So there's a couple of problems with it. Let's say we want to see how many of our T helper cells are Th1 cells, right? In the previous video, we went over what makes a Th1 cell, and one of the big things was the secretion of IL-2, and one of the big things here was, and what creates a Th1 T cell is a macrophage releasing an interleukin 12. So let's say we want to distinguish a macrophage from one that secretes interleukin 12 to one that doesn't secrete interleukin 12. How can we find out whether our macrophage is positive for interleukin 12 in flow cytometry? The big problem there is that cytokines are made to be secreted, right? So they are not sitting inside the cell. As soon as they're created, excuse me, they're pretty much whipped out of the cell, right? So it's a bit of a problem. How are we going to stain the cell for IL-12 if every time it creates IL-12, it releases it? So this is roughly the pathway. The rough ER, the ribosome on the endoplasmic reticulum, will create the cytokine into the lumen of the endoplasmic reticulum. It will then go through the Golgi, and that vesicle will then fuse with the membrane and it's released. This is the canonical secretory pathway of all molecules that are secreted, including cytokines. So what we can do is actually, when we stimulate our cells, so this is mostly when we have cells in isolation, so not in the body of an animal or a human. If we are doing a cell experiment outside the body of an organism, we can add a drug. Now, there's a couple of different drugs here, but we can add 2 different drugs here that stop this pathway in a couple of different locations to prevent the secretion of the cytokine. So we can block the secretion, but not the creation, just the secretion of the cytokine. So what happens in this case is the IL-12 can't get out of the cell, so now it builds up inside of the cell. Now here's the next problem. The next problem is antibodies can't get through membranes. So if we want to stain the interleukin 12, which is inside the cell, how do we get an antibody through that membrane to bind that protein? That's a really tough question. There's a few steps we've got to do. First, we've got to fix the cells. So this is using a fixative agent like paraformaldehyde, for example, which basically causes chemical bonds between everything inside the cell. So now we get all these bonds in between everything inside the cell. It kind of glues everything, so everything has to stay there inside the cell. It's now fixed. There's no movement of proteins in and out of the cell here. So now we can permeabilize the cell, and we typically use a detergent, and this detergent will make the membrane permeable, put holes in it, and essentially prevent it from being a barrier. So now the membrane isn't a barrier, and because we fixed the cell, the interleukin 12 just doesn't flow out of the cell. Like if we permeabilize without fixing, the interleukin 12 or whatever cytokine would just flow out the holes in the membrane. So because we've fixed it, the IL-12 will stay inside the cell when we permeabilize. And now when we put in an antibody solution, that antibody solution can flow into the cell and bind to IL-12. So now we can detect IL-12 here. I made a bit of a mistake here because I thought I was labeling IL-2, but let's say we labeled IL-2, which distinguishes Th1 cells from other T helper cell populations. We might now see a clustering here break off on this TsNE plot, and it might be the IL-2 producing T cells, so these would be the Th1 cells, for example. So you can see cytokine staining is quite an important part of flow cytometry, and so we need to do some extra steps here to be able to get antibodies into the cell to stain them specifically. Yeah, so that was a brief overview of flow cytometry. There's so much I didn't cover. I could honestly yabber on for hours about it. A lot to do with controls and so on and so forth. It's a real art. It is such an art. If you're a researcher, you can't dabble in flow cytometry. You have to commit a good chunk of time to learning the artistry of it. There's a lot of controls there. Some of the fluorophores overlap, and oh boy, it gets complicated. So I just wanted to give you a brief introduction and not dive too much into the crazy details of flow cytometry. So I hope that was a good summary, and now you understand what's going on when you see those flow cytometry graphs in the research articles that you're out there reading on immunology. Cheers, guys.